Hi, this is Miss Litton, and this is my wonderful Period 3 AP Biology class. Say hi.
Hi.
All right. Yeah, hi. First day back from Thanksgiving break, and they are thrilled to be here. So we are continuing on in Chapter 15, Darwin and Evolution, and this portion of our discussion focuses on the evidence of evolution. So just taking a look at these pictures, what do you think this piece of evidence represents? Yeah, fossil. Okay. What do you think this one represents? Biogeographical. Good. And what about this one? What do you think? Anatomical. Good. What about this one? What piece?
Chemical.
Look at that. Biochemical. You have the list in front of you. Oh my gosh. And what about this one?
Molecular.
This is molecular. Biochemical is molecular. Okay, so this development on the whole. Now, development you will see also in anatomical. We talk about that as well. Okay, so these are the big ticket items we're going to discuss. The first one we're going to talk about are fossils. So if you want to be a fossil, you want to grow up and say, I want to be a fossil, you need to be about 10,000 years old. And you need to think about fossils that are found. That's fairly incredible, because normally when things like die, what happens to them? Decompose, or they were what? Eaten and destroyed. So this is something that dies and is still preserved. Not only has it died and been preserved, somebody found it, right? A person stumbled upon it. So that's a big deal right there. And it doesn't have to be actually the death of an organism. It could be traces of an organism. It could be a wormhole through the mud. It could be footprints by an organism. Those are all considered fossils. So there are different ways to preserve things. Things like ice can preserve and make a fossil. What kind of thing might be get in ice? Like a woolly mammoth. Okay. You could use amber. What kind of thing might get a fossilized in amber? Like an insect. You're not going to find a woolly mammoth stuck in amber. Okay. So yeah, Jurassic Park. There you go. Okay. It could be replaced with minerals. Okay, you could have a cast, you could have a mold. So there are different ways that organisms can be preserved, but usually you're only going to preserve parts that are very what? Soft or hard? Hard. Okay, so it has to be something that can be preserved. Not only is it preserved, but it is found. Okay, and discussed. So that's a difficult item. So I gave you some examples here: skeletons, seeds, shells, etc. One argument that's put forward when you look at fossils as a piece of evidence is that there are not transitional fossils. Well, let's talk about what that means. One thing is, think about the evolution of, let's say, us. Okay, probably life started in the water, in the oceans. Who would have emerged from the water? What kind of organism? Amphibians. Who would have evolved from amphibians? Reptiles. Good. Who would have evolved from reptiles? Mammals. Yeah. Yeah. Birds would have evolved also from reptiles and should be classified with reptiles, actually. Okay, so one argument put forth is there aren't enough transitional organisms, but they are out there, keeping in mind you are preserving an organism that's in transition. It's not this or that. Do you see what I'm saying? It's exploiting the middle ground, probably not very adaptive, so probably it's not going to stick around very long. If it doesn't transition and live in this different environment fairly rapidly, it's going to die. Okay? And it has to be preserved and found. So this transitional fossil is a connection between fish and amphibian. This one, between amphibian and reptile, he wanted to see more. Yeah. See more out of the water. Okay, amphibian, reptile. This one, Archaeopteryx, reptile, bird. And birds really should be still in with reptiles. It's just that they were so different, early classification categories separated them out. But really, they should probably still be under the umbrella of reptile. And then therapsids are a mammal-like reptile. So there are transitional forms that are there. So your first piece of evidence, fossils defined, remains and traces of a past life. or any other direct evidence of past life. Transitional fossils resemble two groups that are currently classified separately. And I gave you some examples there. One well-studied example is the history of equus, or horse. Horses started out very, very small little critters, okay? And over time, when you follow the fossil record, they became larger and larger. I'm not talking about those cute little miniature horses that you see in parades, okay? Different, okay? Now, again, this kind of transitions to our next topic about biogeographical evidence, but horses in the environment that they first were developing in were in forests. So in forests, it was good to be small. You have to hide, get around all the little trees, bushes, whatever, hop over things, hide well in the forest. But as the continents are moving, as the area they're spreading out to is in change, as they're competing with one another, some of them probably came out of the forest and they're just on the edge of the forest and moving farther out. It wasn't adapted to be small. You're like looking over them and go, oh, I can get eaten, right? So it's better to be a little bit taller, okay? Wish I was a little bit tall. I need the song to come like this. Wish I was a. There we go. So he was doing it. So you get this slow, gradual evolution, and that where they became taller and taller and larger in order to survive out in the open plains. So that would be a good one to discuss if you're ever going to write an essay about that. Okay? Second piece of evidence, biogeographical evidence. And we have already talked about this, so Slate wants to remind you of it. So go ahead, Slated one.
Okay, Max. Let me tell you a little bit about the biogeography as evidence of evolution.
Let me give you this to help. Keep talking.
And then they started breaking apart.
A bunch of mammals did.
They evolved after the continent. They were all like the same. And then after the bang.
And then we already talked about marsupials being specific. Now, South America had marsupials too, not just Australia. South America had marsupials, but when marsupials and placentals compete, placentals win. Do you know why? Do you know what a marsupial is? Yes. So there's monotremes of your mammals, monotremes, marsupials, and placentals. Monotremes lay eggs. The spiny anteater, duck-billed platypus, those are monotremes. Marsupials, they do not lay eggs, but like monotremes, the development doesn't finish within the mother. Okay? That a mother lays an egg for a monotreme. For a marsupial, they crawl out part way through development, okay, and crawl their way into some sort of indent, cavity, or pocket, and then they have to attach to a nipple in order to survive. Well, that is a very precarious journey, okay? The whole crawling out, whoops, sorry, okay? And so then you're vulnerable during that time. Whereas a placental, you stay in and nutrients are exchanged through a what? Placenta. When you're born, not for us, but like, I was giving this example in period 1, wildebeest. When they're born, like, you better, like, born, okay, run, okay, because something's going to eat you, right? It's not that way for us. It's like they're born, it's like, go. We have this really large head that we can't support, and we're saying, we really value the brain. Hopefully somebody will hold our heads up for us for a while and put a breast in our face because we can't crawl towards it. Somebody will take care of us in that way. And we nurture. We invest in our young in that way. Okay? So marsupials are not just found in Australia, but in the competition with a placental will win. All right. And then endemic species. So this is found at a particular geographic location and nowhere else, like this green vine snake in Sri Lanka.
I want one.
Sri
I'm gonna
Lanka.
buy one.
Found at a certain geographical location and nowhere else. Found at a certain geographical location and nowhere else.
Oh my god.
Okay, what was our first piece of evidence we have discussed so far? Fossils. Okay, what's our second piece?
Biogeography.
What's our third piece going to be? Anatomical. Okay? There are three different, guys, you're very chatty today. Stop it. There are three pieces of anatomical evidence. Yeah, that's exactly what you need to look at for me. Thank you. Start striking him. Don't look over your shoulder. It's you, babe. So there are three pieces of anatomical evidence you want to be familiar with. The first one is homologous structures. Homologous. Like homologous chromosomes, it means the same. Okay? So homologous structures have the same anatomy. For instance, on us, this is our humerus. Radius, ulna. Radius is what comes up from your thumb, is your, okay, then your ulna, carpals, metacarpals, phalanges. Okay? You can find this also in a bat. Humerus, radius, ulna, carpals, metacarpals, phalanges. Their phalanges have webbing between them. Okay, that's exploited in order so they can what? Fly. Look at the bird. Humerus, radius, ulna, carpals, metacarpals, phalanges. Okay, so theirs is built a little bit different. Look at the whale. Humerus, okay, radius, ulna, carpals, phalanges. You can see the same pattern again and again. If our anatomy is the same, then what else must be the same? DNA, and we must share a common ancestor. This is showing you change over time. We use our appendages for picking things up. Somebody else might use it to fly. Somebody else might use it to swim, to walk on. Okay? But we have the same anatomy, which means probably we have a common ancestor that had that anatomy. Okay? So those are homologous structures, not to be confused with analogous structures. Analogous structures have the same function. For instance, both a bird and a bat use what with theirs? Wings. Yeah, wings. What do they do with it? Fly. But bees have wings too, right? Now, bees have wings and they fly, but what is that an example of when we bring bees into the picture? We already learned this. Convergent evolution. Convergent evolution. Okay? You ended up in the same spot because you were dealing with your environment in the similar way. Okay? So I need Blue to please differentiate between homologous and analogous structures. Go ahead.
Homologous structures they all seem like that. But then analogous structures have the same function, but like they don't have a common ancestor. Like homologous, they don't look the same. I'm feeling this.
Yes. Different environments. The first one?
Yeah.
Okay, now, which one of these, homologous or analogous, or both, are used in evolution to show evolutionary relationships? Homologous. Okay? So when you look at structures, you have to be careful. If you're trying to establish a phylogenetic tree, like who evolved from who, you're trying to establish that. You need to watch out for convergent evolution, where you end up and it's basically an analogous structure as a result of convergent evolution. Because when you show relatedness and you show evidence of evolution, you look at similar, the anatomy is the same. And you don't care what they do with it, you just care how it's built. Okay? That's a homologous structure, and that is evidence for evolution. Analogous structure is not evidence for evolution. We're just dealing with that environment in a similar way. All right? So, anatomical evidence, number 1, is homologous structures. Anatomical evidence, number 2, is vestigial structures. These are structures that were useful in your evolutionary past but are no longer useful, so it shows you, it points to a common ancestor. We've talked about this before. Salamanders with vestigial eyes, cave-dwelling salamanders. They don't need their eyes anymore, so they came from an ancestor who had eyes, but because they've lived generation in a dark cave, they don't need them anymore. So any organism that was born without those eyes or damaged eyes had just as much chance at surviving as another organism that had functional eyes. Maybe even better, because maybe the ones that did have the eyes tried to use them where other senses were exploited or developed if you didn't have eyes. You with me? So it would then, that would not be something that determined your fitness, whether or not you had eyes, or had any factor in your fitness, whether or not you had eyes. So it could be withered. You look at the whale, their hip bones and part of a femur. Well, it's not important. It doesn't increase their fitness to have a hip bone or a femur. In fact, it's better off if you don't, because maybe it gave you resistance when you're trying to what? Swim. Okay? So it didn't matter if you had a little bit of it, as long as there wasn't anything that decreased your resistance, you could survive. So you would still see a little bit of femur here and there, right? Because that wouldn't hurt it, only if maybe it did something to affect your ability to swim. An appendix. We have an appendix. What for? So it can burst when we go to the hospital? Why was it ever there in the first place? It just seems like, why would that have ever evolved? What's something that we have a hard time digesting? Alcohol. We have alcohol dehydrogenase. But something else in our regular diet. Hopefully you're not drinking every day. I have trouble, but I drink it anyway. I got it. Okay. What's something we eat all the time? Cellulose. Plant cell walls. We can't digest it because we don't have any cellulase. Okay? But maybe that little pouch housed bacteria that helped us digest plant material. This would be very late in the game because the bulk of the digestion and absorption occurs in the small intestine, and that's coming off your cecum, which is after the small intestine. Did you see that? So it'd be late in the game, but maybe it could still contribute. Okay? So that would be an example of a, vestigial structure. Okay? So piece of evidence 1 for anatomical is what? Homologous. Piece of evidence 2 is vestigial structures. Piece of evidence 3, there we go, possible ancestor to whales. Okay. Piece of evidence 3 is embryological development. When you compare embryos, you can see similarities in their development. And then I added in a little section in your notes just on development. And this takes the development a step further, because what's guiding all of our development? DNA. So when you could study specifically the genes that code for development. That's that last little bit in there. Okay? The genes that code for development. And look, the more similar the actual development, the genes for the development, probably the more related you are. But in the secondary side note, when you look at our development in the womb, it's almost like evolution on fast forward. Because at one point we have gills. At one point we have a tail. So it's almost like you're watching our evolution quickly as we develop. It's like we're still touching base with all those same stages. And the thing is, the genes we have that would code for gills, if we were a fish, code for different things now. In like our pharynx region, in our neck region, and tonsils. Okay? All come from that same region, that same area. The genes are still there. It's just exploited in a different way. Okay, so what was piece of evidence 1 we looked at? No, overall. Fossils. Fossils was evidence 1. I'm not saying it's the most important. I'm just saying in the sequence we discussed. What was piece of evidence 2? Biogeographical. What's 3? Anatomical. And within anatomical, 3 subtopics, you're going to tell me what are they? Homologous structures, vestigial, embryological. Perfect. Okay, let's check our notes on that. Anatomical evidence. Homologous structures, similar anatomy due to common ancestry. Vestigial structures are remnants of structures that served an important function in the organism's ancestors. I gave you examples there. And number 3, developmental structures. Look at the word there, embryological homologies. Blue, explain, why is that a good term, embryological homologies? Go ahead. All right, see, remember, homologies are similar what? Structures, so you're looking at similar structures in the embryo. All right, and then this brings us to our next piece of evidence, biochemical evidence. This is looking specifically at cytochrome C, which, oh, by the way, you know, reductase, cytochrome oxidase. Okay, there you go. Where do you find that? It's in a no. What's in a no? Electron transport chain, specifically of. What name the organelle? Say my name. What is it? Mitochondria, right? Mitochondria. Now, if you study that one protein in mitochondria, who's in charge of moving hydrogens from the matrix to the inner membrane space, right? So it can come back in through ATP synthase complex and make some ATP. Good. If you study that one protein in that membrane, the cristae, right? The folds. What are proteins built out of? Amino acids. So when you take that protein apart and look at its primary structure, sequence of amino acids, when you look at the primary structure and you compare the primary structure against different organisms for that same thing in mitochondria, look right here. We're most similar between what? Us and a monkey. Does that mean we evolved from a monkey? No. It means could we have a common ancestor to a monkey? Yes. Okay. Now, look down here. A pig. That's our next. Of these animals I'm showing you, a pig is still a what? Mammal, right? Still a mammal. But we're more closely related to a monkey than a pig, and then it goes what? Duck, and then turtle, and then fish, okay? And then an insect, etc. Do you see that this shows us like how far back we have evolved from them when our paths split? Okay, so that would give you evidence. See, all organisms share some strong similarities. They all share DNA, RNA, ATP, right? Certain proteins. So you compare those. How similar, how dissimilar are they? If they're very similar, we're probably very closely related. It just so happens when you analyze them, you can see evolution in a nutshell. Okay, so that would be biochemical evidence. And then even further, do you remember how we know the difference between introns and exons? Go ahead, Blue, tell them the difference between introns and exons. Which one was thought to be genetic junk? Introns, because the exons are expressed. So let's pretend we still think it's genetic junk, which some might be. Failed attempts of evolution, etc. If our introns are similar, what does that tell you? We used to have a common ancestor, because this is not, remember, evolution can only work, and natural selection works on a trait that you can select for or against, right? So that's not even a trait that's expressed because it's an intron, but yet it's similar. That must mean we came from a common ancestor because we have the same sequence of nucleotides in our DNA. Do you see that? But now we know that those introns could be coding for RNA, which could then influence the expression of some gene, right? And in developmental biology, we'll talk about homeobox genes and looking at similarities in whether it's a roundworm, an arthropod, a mollusk, and a echinoderm, or something like us, and how we have the same patterns and sequencing. And this section of DNA codes for a head. It doesn't matter what kind of organism you are. And that, again, points to that common ancestry. And in the notes somewhere in there, I have the word homeobox genes. Do I have that somewhere? Yeah. Homeobox genes, and we're going to talk about this later when we discuss development, but there's something you'll already know. You'll already figure it out. Homeobox, and their significance. Homeobox genes code for transcription factors. What do transcription factors do? They help what? RNA polymerase bind to the promoter. Remember, in eukaryotic organisms, it's not so much about negative control, it's about what? Positive control. And homeobox genes code for the transcription factors to help the RNA polymerase bind. So if we have similar transcription factor coding genes, that must mean we have a common what? Ancestor. Okay? So on where am I at? Biochemical evidence. Almost all living organisms use ATP, DNA, and specific enzymes. ATP, DNA, and specific enzymes. It's the same triplet code for the same 20 amino acids. And we have similar introns. And developmental biology evidence, similar set of regulatory gene. And then that's where I have the homeobox. Hox genes. All right. Now, so when you see something like this, now Google for me, how many species of beetle are there? Because I'm pretty sure we don't have them all on this page. How many different species of beetle are there out there? How many? 350,000. So I'm short just a tad. You should now be able to explain why there are 350,000 different species of just beetle. Okay? I'm not telling you not to believe in God. You know that. I'm not telling you that, right? Okay? I'm not saying that. I'm just saying, do you think, even if you do believe in God, you know I was raised in the church, right? So I've heard every argument, really bad ones, really, really bad ones that are taking my notes and getting mad at my pastor. I'm like, that is not science. But did God create 350,000 different species of beetle and 500,000 different species of plant or whatever we're at right now? Or is it possible that they could have had overproduction of young? Finish these sentences for me. Overproduction of young. And of that overproduction of young, there were what? Yeah, there's going to be competition, and there's going to be what between them? Variations. And it has to be heritable. It has to be in their DNA. And there's going to be a what? Competition. There's going to be a struggle. And those who have the best variations, the best adaptations, those are ones who are going to what? Survive and reproduce, and you have lots of time. That's natural selection. That's why you have so many beetles. Okay? That would be the big picture. So riddle me this. Sign in as yourselves.
Max? Thanks.
What's their name? Yourself. But for some reason, I just froze again. Oh, spinning wheel of death.
Thank you, Max.
Whoa. I don't know what just happened there, but let's try again.
Yeah.
Oh, it's freaking out. I can't sign in. I can't do anything. It just like froze.
I don't mind that though.
Okay. Well, we don't have the questions in this presentation, but we hope that was super helpful. And if you're studying late at night, we hope you have a piece of toast. Make good choices.